Yes, good afternoon, students. Hope I'm audible. Yes, okay, very good. Fine. We shall continue now. So, we are in the last part of our topic, the last topic which is remaining for us. If you are able to guess what it will be. See, you might have learned about heredity. Some knowledge at least you will have about heredity, correct? We acquire certain characteristics from our parents, no? Like some similarities we have, no, with our parents. That's because of the genetics, that is because of the genes which are being transferred from our parents to us. And our parents have some similarity with their parents, correct? Some similarities are there, some characteristics are similar, some of the traits we carry from them. Even our some of the features might be similar to our grandparents also. So now why is this happening? Like, what is that one thing which is responsible for the carry forwarding of these genetic characteristics? Genetics is the whole branch of biology, which studies about this variation, and where you are studying about that heredity, that is called as genetics. Now my question to you all is, what is that thing which is responsible for these, what is being carried forward? Now if you want to check that whether the child belongs to that parent, what do they check usually in the lab? If they have to check, DNA, correct. DNA, no, this you might have seen in all the, most of the time it is in our Hindi serials and the movies. If they want to check that the child belongs to that particular parent, first thing what they will check is the DNA, correct? No, DNA check will be done. So DNA is standing for deoxyribonucleic acid, okay, which is responsible for the transmission of the heredity character. Okay, so here in detail you'll be studying about, it is one part of it. Okay, so in detail what you are going to learn here is about nucleic acid. So one form of nucleic acid is DNA. Okay, now where is this present? You have learned about cells in, and you know that inside the cell we have nucleus. Inside the nucleus, we are having these thread-like structure. Chromatin fibers are present. Okay, so chromatin fibers are present, and then when the reproduction is going to take place, when the transmission is going to take place, see, they convert themselves. These thread-like structures are there. No, they convert themselves in this form, chromosome form. And human beings, they have 23 pairs of chromosomes, right? 23 pairs of chromosomes we have within us. So that 23 pairs will be transferred from the parent only. We are diploid in nature because when the embryo has to be formed, it will be 23 which we get from our father and 23 which we get from our mother. Okay. So then on the whole, we will be having 23 pairs of chromosomes. So these chromosomes will be formed during the reproduction time, and that chromosome is consisting, it's a coil like structure. You can see here how much coiling has taken place here. If you start opening it up, you will be able to see this double stranded structure, which is DNA. Okay, so DNA replication helps us to transmit that character from one generation to another generation. So now you are going to learn about these things, about nucleic acid. What is the, for us important is not the biological aspect of it. For us important is what are the chemical components present in this. Okay, what are the chemical components which are there in nucleic acid? So let's go in detail about these. So basically, nucleic acid, I have explained you. It is responsible for the heredity character called as chromosomes, and those chromosomes are biological molecules which are made up of DNA. Finally, it is having those things. Okay. Now, there are two kind of nucleic acid. One is DNA and the other one is RNA. DNA and RNA. There is a full name for this. This DNA and RNA stands for, DNA is deoxyribonucleic acid. It's given here, see. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. That's because of the chemical component present over there, the name has fallen like that. Let's see what are those. Okay, let's go into detail. So two components, remember, there are two components for nucleic acid, that is DNA, and then we are having RNA. Nucleic acid is a polymer. Okay, it's a polymer of nucleotides. You can see here the huge molecular structure I have put here. Okay, you can see the different components. You can see here the P group which is written. I'll explain what and all is that. Okay, so you can see this P1, you can see a sugar attached on this side, and then this side you can see one more group like this, which are held to one another through a bonding. Okay, but then this T part is there, no, that's one more group. T is there, one sugar part you can see, and one P part is there. So that same thing which is getting repeated one after the other, giving rise to a huge molecular structure, that's the polymer, and this huge molecule is called as nucleotide. So nucleic acid is a long chain polymer of, it's a long chain polymer of nucleotide. Okay, so let's see what are nucleotides here. What is that nucleotide consisting of? So we'll still go into detail of that. So composition of nucleic acid I've put here. So that nucleic acid, I said it's a polymer of nucleotide. That nucleotide consists of two things. One is nucleoside, SID, nucleoside, and the another one is the phosphate group. That nucleoside again has two parts in them. One is the base, okay, that is nitrogenous base which is present, because it's a protein only. Finally, it's a protein molecule, so they will have nitrogen containing compounds there. So nitrogenous base is present, and then we are having the sugar. So when base and sugar combine together, when nitrogen is base, it is nitrogenous base. Okay, when this nitrogenous base combine with the sugar, you get that small dimer which is formed, that is nucleoside. When nucleoside combines, I'm going in the reverse order now. Okay, when base, nitrogenous base combines with the sugar, what you get is, I said, nucleoside. When nucleoside and phosphate combine together, what you get is nucleotide. When many nucleotides combine together, we get nucleic acid. Is this followed? The components are clear. Components are important for you. Okay, how is nucleotide formed? What are nucleotides containing? What is nucleoside? So now if you see the structure, you'll be able to understand. See, I'll go back to that structure. You can see here that P part I said, that's the phosphate group. This P1 is there, no? This blue color one. So that is a phosphate group. So here we have that phosphate group. And then you can see the sugar part here. So here we are having that carbohydrate part. And this side, this T1, this T, I'll circle it here. This is that nitrogenous base. Okay, this is our nitrogenous base. I'll write NB, nitrogenous base. So when nitrogenous base combines with the sugar, when these two are combining, only these two if they are present, that is your nucleoside, SID. When this nucleoside combines with that phosphate again, when all the three components are combined together, that give rise to a nucleotide. Okay, when many nucleotide combines, you get a nucleic acid. So nucleic acid, I said, it's a long chain polymer. So that again I have just put over here, this side. You can see nucleoside is when nitrogenous base is combining with a pentose sugar. But when all of these three are combined together, that is nitrogenous base, pentose sugar, and one phosphate group, when they are linked, these three components together give rise to a nucleotide. So if you hydrolyze this, and chemically you try to break them down, you will get three parts, no? You will get nitrogenous base present here, you will get a pentose sugar, and then you will end up getting one more group, that is phosphate group on hydrolysis. So these are the three components. Okay, let's again go into detail of these, the chemical composition of these. So we said one base is there and a sugar is there. Okay, so on complete hydrolysis of your DNA or RNA, rather anything is composed of the same it is. Okay, the only thing is DNA is a double-stranded structure. DNA is double-stranded and your RNA is just single-stranded. But they are composed of the same thing. So you can see here, I said they are finally, when you break them down, they will give you one pentose sugar, they will give you one phosphate group, and they will give you one nitrogenous base. Okay, so nitrogenous base are present. So let's see how is that pentose sugar and what are those pentose sugar. So in case of RNA, it is beta D ribose sugar. You should know the structure, students, here. You should remember this structure, beta D ribose sugar. See the structure, how it is. The fourth carbon and third carbon position is similar to fructose, but these two are different. Beta you can remember. Beta means OH is up and H is down. Correct. No, beta means OH is up and H is down. Only this position you have to remember. Or you can remember third and fourth, the second and third position, it is same. HOH and HOH. And fourth position, it is similar to fructose. Beta you can remember because beta means OH has to be up and H has to be down. So beta D ribose. The next one is the DNase sugar group which we are talking about. DNA, it is called as deoxyribose. Okay, it is called as deoxyribose and it is 2-deoxyribose. Second position, it is oxygenated. Deoxygenated means that oxygen over there is missing. So you see that second position, compare that ribose second position. This is our second position where OH is present. And you see the second position of DNase sugar. There is a no OH, O present, that O which is there, that is missing. Only H is present. Okay, so it is called as deoxyribose. That deoxyribonucleic acid, when we are seeing that deoxy stands for because second position there is missing of oxygen. Okay, so structure you have to remember second and third position HOH, HOH. First position, since it is beta, you can remember OH is up and H is down. The last position is similar to fructose. Okay, it is pentose only, pentose sugar, it is 5 carbon are there in that. Okay, then moving on with the next. This is about the pentose sugar. Deoxy, you remember second position, there is no oxygen. Okay, deoxy because second position. If you remember ribose, you can write deoxy. It will be easy. Second position. Okay, oxygen is eliminated. You have to remove the oxygen from here. Okay, next coming to that nitrogenous base. These are the nitrogenous base which we are having in DNA and in RNA. See the difference and the similarities. In DNA, we have adenine. Only names are enough. Structures are not important. So just remember the names. It is adenine, guanine, cytosine and thymine. Adenine, guanine, cytosine and thymine. AGCT. AGCT. And in RNA, you see, RNA, the nitrogenous base present over here is adenine. It's adenine, guanine, cytosine, and then you are having uracil. So what is the similar nitrogenous base which are present? These are similar. See, AGC and AGC. This also they ask. What are the similar nitrogenous base which is present in RNA and DNA? Similarity is this. Okay, these are similar. Only the difference between them is this group. That is thymine and the other one is uracil. So in DNA we have thymine and in RNA we are having uracil. Okay. And difference in the group also they can ask. Which is the nitrogenous base present in RNA other than the similar ones. Uracil is the one and in DNA it is thymine. Okay. So AGCT here and here AGCU. These are the structures, you see. And they are categorized based on their structures. They are categorized into two. That is purine. One is called as purines and the other one is called as pyrimidine. That's because of the structure they have, no? So they are named like that. Purines are adenine and guanine is purine. And thymine, cytosine and uracil are pyrimidine. That's because of the ring, pyrimidine ring they are having. So it is called as pyrimidine. So thymine, cytosine, uracil are pyrimidine bases and adenine and guanine are purine bases. Okay, adenine and guanine are purine bases. Thymine, cytosine and uracil are pyrimidine bases. Structures, again I said it's not important for you, but at least name you should know. Name you should be familiar with. Now let's see how the structure is. In DNA and in RNA, how that structures are arranged, you should know only that much. Okay. So in nucleoside, this nucleoside is formed by attachment of nitrogenous base at first position of the sugar. First position of the sugar, you will find a nitrogenous base attached. And that is enough for you. Okay. And second one, you see in nucleoside, The phosphorus group which is there, that phosphoric acid group is present at fifth position of the nucleotide. So you will see that. And between these two nucleotide chain, they are arranged in a parallel, anti-parallel way. If one is going in this direction, the other one will be arranged in the another direction, opposite direction. They are not arranged. This is how that DNA structure is. Okay, see. First carbon is here. I'll just label it. First carbon of the pentose sugar. This is the first carbon, C1 carbon. To the C1 carbon, you can see the base is attached. You can see here. This base, nitrogenous base, is attached to the first carbon. And fifth carbon, see 1, 2, 3, 4. This is fourth one. And this is your fifth carbon. To the fifth carbon, you can see that phosphate. Phosphate group is attached. Phosphoric acid is attached over here. Okay, so this you should remember. These two are attached to one another. Nitrogenous base is attached to the first carbon and the phosphate group is attached to the fifth carbon. So like that, these two, when phosphate group reacts with the third position, see third position of this, when they react with one another, they end up forming that nucleotide chain. Okay, so this will be dinucleotide which is formed here. You can see in front it has been formed through this phosphorus linkage. It is called as phosphodiester linkage because the group is like that. Phosphodiester, ester is having C double bond OR. No, you can see here phosphorus is giving you that P double bond O, and then R will be this group like that. So like that, the similar groups are formed. It is called as phosphodiester linkage. So one nucleotide is held to another nucleotide by this phosphodiester linkage. In saccharides, it is a glycosidic linkage. In protein, you remember it is a peptide linkage. Okay, so linkages, at least keep in mind what kind of linkage each and every polymer over here is having. So one after the other, they are linked to one another through a phosphodiester linkage. So like this, it continues. When many molecules react like that, see fifth position and third position, fifth position and third position, like that they are reacting. So like that, anti-parallel, one more strand will be there, anti-parallel, which will be running in this direction, and they are held to one another through a hydrogen bonding. Okay, so this is the same thing which I told you. When many molecules combine together, you get a polynucleotide. So this is for polynucleotide. This one which I have shown is just a dinucleotide. I just showed one condensation. Here many condensation has taken place. And nitrogenous base here present are different. So this one chain from 3 prime, see here, third position, no, it is reacting. Third position is where the linking is happening. And to fifth position. So third, 3, it is called as 3 prime to 5 prime. It is called as 3 prime to 5 prime because here is where the linking is happening. And another chain will come from this side. 3 prime to 5 prime from this side, and they will start bonding with one another. Okay, like this. See here. From this side one chain is going. You are able to see here. This side one chain is moving, and the another side the chain is moving in the opposite direction. So these bases which are present, they have nitro NH group is present there. C double bond O groups are present. So they will be held to one another through a hydrogen bonding. So if we have our DNA, that's basically because of the hydrogen bonding. So if DNA exists in that double stranded form, it's because of that chemistry is important more than biology here. The chemical composition makes them, okay, in that particular structure, that double helical structure which was given by Watson and Crick. These are the two scientists who got Nobel Prize for working on this DNA double helical structure. They produced that structure, but more work was done by some other scientist also. Okay, so here adenine and you see the bonding here. You can see adenine is binding with thymine. Adenine is forming a bond with thymine, and only cytosine is forming a bond with guanine. It is always like that. Adenine can only have a hydrogen bonding with thymine, and cytosine can have only a bonding with the guanine. And whenever adenine forms a bonding with thymine, it's a double bond. And whenever guanine forms a bond with cytosine, it is with a triple bond. Okay, so here, this is how the DNA structures are held to one another. Did I make it clear, students? Did you follow? Like, there are too many things. I understand that. But then, did you understand what are the things which are present here and how they are held to one another? I'll repeat this once again. Once quick revision for you. See, I'll tell you the important things which you need to keep in mind. Very important things which you need to keep in mind. Just listen once again. I know you are a computer science students. You all don't have any base knowledge about biology. This is more about biology and the bonding which is happening here. Okay, so we'll start with nucleic acid. So in nucleic acid, in simple language, I'll tell you, two kinds of nucleic acid you are studying. One is RNA and the other one is DNA. Okay, one is RNA and the other one is DNA. You can see here. Let's go for the components of this. I'll directly go for the component. So this nucleic acid, basically, it's a polymer. It is polymer of nucleotides. Okay, nucleotides are made up of, I said nucleoside and phosphate, and that nucleoside is made up of these nitrogenous base. So let's start with this first. See, if you take nitrogenous base and combine with the sugar group, if nitrogenous base combines with the sugar, what you get is called as nucleoside. This is called as nucleoside. Okay. When nucleoside is made to react with your phosphate group, when this nucleoside reacts with the phosphate, what you get is nucleotide here. I am just putting an arrow mark that gives you nucleotide. When many nucleotides combine together, imagine when many nucleotides are combining together, one more nucleotide here, which will have all these three components should be there in them. When many nucleotide combines, you are getting polymer of nucleic acid. This is understood. Simple language what I said. Three component you should remember. You will have to remember this. You have to memorize this because it is more of biological term and all that. There is a base, I said, that is nitrogenous base. When they combine with the sugar, they form that smallest compound formed is called as nucleoside. When nucleoside combines with the phosphate group, it is giving you nucleotide. When many nucleotides combine, that is giving you nucleic acid, which is your DNA and RNA. Okay, which is our DNA and RNA, which is present inside our body, inside our cell, all these are present. You're just studying about it. These are already there. Okay. So if you see the structure wise, this is how it will be. See, nitrogen is base with one pentose sugar. That's nucleoside. When all these three are combined together, that is giving you nucleotide. Followed, no? Then I went little more in detail. What are the pentose sugar? What is that nitrogenous base? And the phosphate group. This much is clear. I'll be asking, you all just respond. This is understood. Three components present over there. Okay, fine. Okay, now we'll move on with what are the other components which are there. So in DNA, if you hydrolyze this, I said, if you hydrolyze this, you will be getting three parts. One is nitrogenous base, one will be your pentose sugar, like final breaking down of all the bonds. The small compound what you will get will be one is nitrogenous base, one is your pentose sugar, one more will be your phosphate group. One more group which will be formed will be a phosphate group. These three components will be formed. So what is that pentose sugar and that nitrogenous base we are studying in detail. Okay. So we'll go for that. So in DNA and RNA, DNA which is consisting of these three parts, that is pentose sugar, phosphoric acid and nitrogenous base. In that pentose sugar, let's go for that pentose sugar. What is that pentose sugar? That pentose sugar is beta D ribose. It is beta D ribose for RNA. Okay, for RNA it is beta D ribose. If you want, you write it down. So then it will be easier for you. And in DNA, the sugar group is there. No, it's also called as sugar moiety. That small sugar group which is there. The sugar group which is there, it is beta D ribose for RNA. R is for that ribose group only. And DNA, it is deoxyribose. That D is for deoxyribose here. So structure wise, how it is? See, first carbon has one OH and H. That's because of beta position. You know, in beta, OH will be up and H will be down. Second and third position, you draw it similar. H, H and then OH, OH down. H and H up, OH and OH is down. And in the fourth position, the carbon is having CH2OH H up and one H is down. Okay. It is similar to fructose. That fifth position, okay, it is similar to the fructose. Only this second and third position you will have to remember. Now in deoxyribose, the only change over here is in the second position. So it is called as 2-deoxyribose. Second position, that oxygen is missing. That oxygen is not there. Okay, for DNA, sugar moiety, they don't have that oxygen present. It is just the hydrogen. Otherwise, the structure is similar. Okay, sugar structures is understood. Structure you will have to remember. Okay, for that, we can't do anything now. Only the difference you should understand now. Yes, no? So you understood that sugar is beta D ribose and 2 beta deoxyribose. You understood the difference between the structure also. Okay, let's go for that nitrogenous base now. Pentose sugar we covered. Just now what we covered was pentose sugar part. Now we'll go for that nitrogenous base, the second part of it. Last we are attaching the phosphoric acid group. That will be a third part. So the second part is our nitrogenous base. These are the nitrogenous base which are present. Total 5 are there. In that 3 is common for both DNA as well as RNA. That is adenine, guanine and cytosine. You will have to remember this name. Adenine, guanine and cytosine. That is for DNA. Okay, with DNA and RNA which is similar. The nitrogenous base which is similar for DNA as well as for RNA are these adenine, guanine and cytosine. Only the nitrogenous base which is present different in both the case is this, the last one. Thymine is there in DNA and uracil is there in RNA. So if they ask you which group is not there in RNA, it is thymine. Which group is not there in DNA, it is uracil. Which are the nitrogenous base which is similar in both of them? It should be adenine, guanine and cytosine. This is followed now. Nitrogenous base. Yes, no? Shall I proceed? Then further they have just classified them based on their structure. They are called as purine bases and they are also called as pyrimidine base. If you can, you remember this. If you can't at all, then leave this. Okay, the one who wants to score well, no students, I expect you to remember this. Others, if 1 and 2 marks is not important for you, you can skip this. So purine bases are this, adenine and guanine. Adenine and guanine are categorized under purine. That's I said because of the structure they are having. Purine, they are called as purine, and the thymine, cytosine and uracil, they are called as pyrimidine base because of the structure again. Okay, so these three, pyrimidine, those two, adenine and guanine, are purine bases. Okay, then I said how they are arranged. That's it. Then over. Then that is the whole thing about the chemical composition of nucleic acid. So how are these arranged? I was explaining you. If you see, 3 groups are present. No, nitrogenous base we are having. Then we have that sugar group. And then we have that phosphoric acid. I am bringing it here now. That phosphate group which is present. So if you start from the first carbon will have your nitrogenous base. Okay, nitrogen base will be attached to the first carbon. And then here, 5th carbon which is there, see if you number them, you understand. This is first, this is your second, this is the third one. Here is your 4th carbon and this is your 5th carbon. 5th carbon has that phosphoric acid attached. Okay, so that phosphoric acid is here. Do not remember the structure. Don't worry about the structure. They don't ask you the structure. Okay, if you can just write the simple version of it is enough. If you just write, see, this is how the pentose sugar is, no? Just draw that pentagon, one oxygen you draw here. Just write here, what is that? Nitrogenous base. Just a nitrogenous base. That is in first position. That is in first position. Okay. Then you have second, third position. This is your 4th position. Here is your carbon. This carbon which is in the 5th position, is having a phosphate group, P group, over. So altogether, these are your nucleotide. This whole thing is your nucleotide. Now that phosphate group is there, that will be linked to another nucleotide. But which position? Third position. You can see here, no? This is visible to you. When they link with one another, they are undergoing condensation with elimination of water molecule, H and then OH. And they link to one another through this oxygen. So third position and fifth position are held by phosphodiester linkage. This linkage is called as phosphodiester linkage. And you are getting this dimer here, dimer. Like that, when many molecules combine, see here, many molecules are combining here. 1, 2, 3, 4. This is our fifth position. Fifth position and then again third position. Again here fifth position is combining with third position. Again this fifth with the third. So like that, continuously they are forming a polynucleotide. Understood now? What is polynucleotide? This polynucleotide itself is nucleic acid, no? This itself is nucleic acid. Okay. Finally, whatever you are getting. So in RNA, it will be only one stranded. You will have only one strand attached like this. But in DNA, I said, they will be having a two strand. Double strand will be there. So two strands will be there. One strand moving from that 3' means that third position to fifth position. One more will be moving from anti-parallel direction, third position to fifth position. And both of these two strands will be held to one another, I said, by a hydrogen bonding, where adenine will bind with thymine and cytosine will bind with guanine through hydrogen bonding. Whenever adenine binds with thymine, I said there will be a double bond. And when cytosine binds with guanine, it is with triple bond. This much only you are supposed to remember. Now at least you all know what is there in this nucleic acid part. I know remembering the name, it will take little time for you. That's okay, you will be able to remember later on. Once or twice when you revise, you will be able to remember. Yes, no? Understood? That's all about the nucleic acid part. Okay, that's all about the whole thing. This much you should study. Now, shall I ask you a question? You will be able to answer now. Let me see if you are able to answer me. I have not displayed any question like that. I'll just ask you or listen to me. I am expecting the answer from you. Now tell me, the base which is present in DNA, the base which is present in DNA, but not in RNA, but not in RNA. If you want, you can write down this also. Others can write it down later. Answer this question. The base which is present, I'll give all the questions which can be asked in your exam. The base, first question is this. Name the base which is present in DNA but not in RNA. What is that? Which base is that? Quickly reply, everyone. Base which is present in DNA. It is there in DNA, but it is not there in RNA. Yes, very good. Thymine is the answer. Thymine is very good. Correct. Thymine is the answer. Thymine is the correct answer. Thymine is there only in DNA, no? It's not there in RNA. What if a reverse question is asked? Name the base which is present in RNA but not in DNA. That I'll write here. Base which is present in RNA but not in DNA. But not in DNA. Yes. For that answer is uracil. Very good. Correct. For that answer is uracil. Now, what is a nucleoside? Next question for you. What is a nucleoside? Third one. What is nucleoside? Just with the two words, if you tell me, is enough. The letters are also fine. What do they contain? What is a nucleoside? It consists of nucleoside is, what are the two things which are present? Yes, very good, correct. Base and a sugar. One nitrogenous base is there and one pentose sugar is there. So they consist of a nitrogenous, it consists of a nitrogenous base and one pentose sugar. If you want, you can mention the position also. In the first position of the pentose sugar, nitrogenous base is attached, correct? What is the pentose sugar which is present in DNA? The next question for you. Which is that pentose sugar? Fourth one. You have to name. Name the pentose sugar which is present in DNA. You have to name that. Give the full name. Pentose sugar which is present in DNA. Yeah. What ribose is it? That full name, beta D deoxyribose, no? DNA it is. It is DNA. Beta D and then deoxyribose. Yes, beta D then deoxyribose. Similarly with RNA, it will be just beta D ribose it will be. Okay, beta D ribose. Now tell me, what is that linkage which is present between the two nucleotides? How dinucleotides are linked to one another? Yeah, beta 2-deoxy also. Yeah, it is better if you mention 2-deoxy also. Yes, correct. Beta D 2-deoxyribose. Because you are mentioning that second position, that oxygen group is removed. Yeah, it's better if you mention. Now tell me, what is that linkage which connects two nucleotide? The linkage between, what is that linkage between dinucleotide? Even with polynucleotide, it is same. Dinucleotide. What is that linkage which is present between them? Go back to this ester. Which ester bond was that? This, no? See, this bond I told you. Phosphodiester bond. Okay, so phosphodiester linkage it is. Yes, very good. Correct. So you can write the answer. It is two nucleotides if they ask you, or when many nucleotides are joined together, the linkage which joins them, they are joined together by phosphodiester linkage. Phosphodiester linkage. Now, how many bonds? This also, one more thing, which speaks about the number of bonds. How many hydrogen bonds are present between adenine and thymine? How many hydrogen bonds? I'm just writing HB, hydrogen bonds it is. Okay. HB stands for how many hydrogen bonds are present. It is present between, one is, you should know, that is adenine and thymine. So one is between adenine and thymine. Second one you should know between guanine and cytosine. You should know it is between guanine and cytosine. Yes, very good, correct. So between adenine and thymine there is a double bond, correct. Two bonds are present. So adenine and thymine are held to one another by a double bond, and cytosine and guanine are held to each other by a triple bond. Oh no, much questions they won't ask here. And ribose sugar drawing, you remember, I am not asking that. I know it is difficult for you to answer that. So that structure, how to write, you remember. And which are the similar groups which are present in both of the DNA and RNA? See, there is no 3 mark as such. That 3 will be having a sub question. It won't be complete 3 mark question. It will have 2 questions there combined. Getting? It won't be like that. Okay, so that 5 marks they are saying, in part D they will be asking. So that 5 marks won't be just like that. It won't be just a 3 and 2. That 3 mark will have a sub question also. Maybe they will ask you why sucrose is called as reducing sugar. Then give one example for that. Another molecule which is having no, like that, some like combined questions will be there. Or it can be like, what is peptide bond and how are they formed? What is dipeptide and how many peptide linkage are present in tetrapeptide? See, like this combined question, which can be asked for 3 marks. Okay, so this chapter gets over here. So what I want you to do is, now I'll just end this session, take 5 minutes break, and then rejoin once again for the next class. Okay. I'll continue. I'll start with the new chapter, the last chapter which is remaining for us. So this chapter is done here. So we will wind up. Now, I just wanted to know, do you want me to do a revision of chemistry in everyday life? Because you people have done it, the seminar, and you have done it well also. But do you want me to do it once again? You want me to revise it once again? Yes. Okay. Fine, fine. Okay, that will be done then. That will be done later at the end. So I'll finish the chapter, the major chapter which is remaining. I'll finish that first, and then I'll do that chemistry in everyday life. That will be at the end. Okay, but I'll be just briefing through because you have done in the class. I'll just tell you what are important and where you are supposed to focus. Yes, now I'll end this session will get over now. Okay, it will just get over now. So take few minutes break and then rejoin once again.